Our planet is a living planet. Life is everywhere. In the air, on land, under the ocean, at the microscopic level, and the macroscopic. It's breathtaking. And making up 3 groups of cellular organisms: Eukarya, Archaea, and Bacteria. These are the 3 domains of life. All cellular organisms descended from Leuca, the last universal common ancestor of life. Leuca was probably a mega-organism, a loose collection of cells without their own identity. Let's talk about how domains of life originated from Leuca. A number of competing scenarios describe the evolution of cells. They generally explain phylogenetic trees or networks built from genomic data. For example, a phylogenetic network of all known molecular functions of organisms supports a 3-domain of life scenario of cellular diversification, in which there are 3 clearly separated groups of organisms. The canonical view is that there are 3 distinct domains of life: Archaea, Bacteria, and Eukarya. This canonical view also states that bacteria is the first domain of life to appear in evolution. This view is supported by phylogenetic analyses of the sequences of universal genes. An alternative view is the eocyte hypothesis. Here, Eukarya diversifies from within Archaea. Eukaryotic organisms are therefore considered modified archaea, and the living world is effectively made of 2 domains. This view is supported by phylogenetic analysis of concatenated sequences of universal genes. A third view is the fusion, or ring of life 3-domain scenario. In this hypothesis, a bacterium fuses with an archaeon to form the eukaryotic cell. These chimeric origins are supported by blast analysis and phylogenetic networks. Here, we present the Archaea-First scenario, in which Archaea is the first domain to appear in evolution. Eukarya and bacteria diversify later. This view is supported by phylogenomic analysis of molecular structures and functions. Archaea-First has considerable explanatory power, but has been an often neglected view of diversification of life. First, the Archaea-First scenario parsimoniously explains the membrane phospholipid makeup of cells. In Archaea, isoprenoid side chains are typically ether-linked to glycerol phosphate. In contrast, in bacteria and eukarya, fatty acid chains are ester-linked to a different enantiomer of glycerol phosphate. While the Archaea-First scenario only requires a single origin of the enzymes making up the lipid biosynthetic pathways, the other scenarios require many more. Second, the Archaea-First scenario parsimoniously explains the origins of the nucleus, a membrane that encloses the genetic materials of the cells. The nuclear commonality hypothesis posits that eukarya and some lineages of bacteria, the PVC bacteria, share an endomembrane nuclear structure with nuclear pores, which derives from a common ancestor. This hypothesis is compatible with the Archaea-First scenario, but not with the canonical 3-domain and eocyte 2-domain scenarios, which require multiple origins of the nucleus. Structural phylogenomic analysis strongly supports the Archaea-First hypothesis. Proteins are made of component parts. These are the protein domains, which are the structural, functional, and evolutionary units that hold deep historical information. A Venn diagram describing how domains of life share domain families reveal a substantial number of universal families. This fact indicates protein structures hold a strong memory of evolutionary history. To study the origin and evolution of protein domains, a rooted phylogenetic tree of domains is constructed and used to assign an age to each domain family. These ages are then used to build a timeline of domain families which reveal the evolutionary accumulation of domains unique or shared by domains of life. The timeline can be divided in 5 phases. The first phase is defined by the initial accumulation of universal families. The second phase shows how accumulating families are lost in Archaea. The third phase shows the appearance of the first bacteria-specific families and families shared by Archaea and bacteria. The fourth phase shows the first Archaea-specific and Eukarya-specific families, appearing together with families shared by Archaea and Eukarya. The accumulation of domain families could be mapped onto a tree of life with basal reticulation. This mapping generates a timeline of trees of life, which remarkably unfolds the Archaea-First diversification scenario. In this timeline, green and purple arrowheads describe coevolution and horizontal sharing between Archaea and the other 2 domains of life, bacteria and Eukarya. The AB and AE Venn groups are examples of sharing of protein domains between domains of life and suggest significant horizontal exchange of genetic information and coevolution of organisms. This sharing of domain families was significantly biased towards one of the 2 main groups of archaeal organisms. Bacteria shared many more domain families with Eukarya. This bias supports the co-evolutionary hypothesis that emerging Eukarya interacted with bacteria while emerging Archaea interacted with Eukarya. This important theory of co-evolutionary diversification now demands further exploration. We propose the use of molecular structure to study diversification in deep branches of the tree of life and molecular sequences to study recent diversification events. The history of life in the living planet must be uncovered.